Hi, good morning, everybody. So I have good news and great news. So the good news is that in this Tuesday, we are not going to have in-person class, just need to see my lecture. The best news is that because of that, I'm going to give full attendance to everybody. So hooray! So today we're going to be talking about bacterial gene regulation. And we have to remember that gene expression is a multi-step process that involves transcription and translation. So as you can see here, we go from a gene that's going to be transcribed into an mRNA, then translated into a polypeptide that's going to be folded into a protein that's going to have a function. Okay, we do have a regulation of how much of a gene should be expressed. Some genes, they are unregulated. These genes that are unregulated means that they are always expressing, they have a constant flow of production of protein. We call these genes constitutive genes, while for all the other genes that they require sometimes to be produced more, sometimes to be produced less, we're going to have a regulation for this. Okay, and we have to remember, does it cost energy to make a protein? Think about it, guys. We, if you guys remember, it requires at least one ATP and one GTP in the process of translating, and then in the process of translating later, transcribing and of transcription and then translation. Okay, so yes, it costs energy to produce. Also, we're going to use a bunch of nucleotides and bases that are there to produce these mRNAs and amino acids and all that. So yes, it costs, and because of the costly, because it costs energy to produce it, it's better just produce when it needs, when it requires. So one way of regulating is through regulating the rate of transcription, okay? And that's the most common way of regulating a gene expression is through regulating how much you want to this gene to be transcribed. And that's the best way to do it. In general, most genes are always being expressed, but it can be into a lower or sorry, a slower rate or a faster rate. So if a transcription is regulated by a regulatory protein that binds to a DNA, and this is gonna make the increase the transcription, we call this regulatory protein as an activator, and we call this regulation as a positive control. While if this protein is gonna inhibit the transcription, we call this protein as a repressor protein or repressor regulatory protein. And in that way, you're gonna have what's called a negative control. Okay. And you're gonna have some examples of gene expressions through our class, and you're gonna see that some of them they're gonna be through positive control, some are gonna be through negative control, some genes are only expressed when they have too much of something, some other genes that are not only expressed when they have too few of something. Okay. These regulatory proteins, they usually have two main domains. One where it binds to the DNA, and another one that binds to what's called an effector molecule. That's also going to regulate if that protein can bind with the DNA or not. Okay, so here's some example. So we can have an effector molecule that can be an inducer. It means that it's going to increase the rate of transcription, and it can increase this rate of transcription by two different ways. The first way is by when it connects to the activator protein, this activator protein is going to start the process of transcription. We call this a method of inducing a inducing the gene expression. Okay. Another way that can do it is by when the inducer connects to a protein, this protein that is there to repress the production of that gene no longer is going to repress the production of that gene. That way, it's also inducing the gene expression. Okay. We say that an inducible gene is a gene regulated by an inducer. So we have some genes, you're going to see some examples in the future slides, where we show this process in action. On the other hand, we also have effector molecules that are going to repress the production of repress the gene expression of a certain gene. So for example, we have a effector molecule called corpressor that's going to decrease the rate of transcription by binding into a repressor protein. So we're going to have a repressor protein that's in just around, and when the corpressor binds into the repressor protein, this repressor protein is going to be activated, and this is going to make harder for the RNA polymerase, or make impossible for the RNA polymerase to be activated. That way, you're going to be repressing that gene expression. You also have effector molecules that straight up are inhibitors. So it's going to connect into an activator protein. And when connects to this activator protein, this protein no longer activates the gene expression. Okay? These genes that are regulated by corpressors and inhibitors, we call this gene a repressible gene. So we have an inducible gene and we have repressible genes. Okay? So let's see some examples here. So one very important example that you can see is one that's very, very common. It's well studied as well. That's it's from already like 70-year-old experiment with E. coli. So in general, E. coli grows in common media, like agar media, that's very bland and have almost no nutrient. It's very poor in nutrients, but it has glucose in there. And when you see the expression of enzymes that produce that can digest lactose, is usually very low to non-existent. So in general, where E. coli usually grows, the bacteria doesn't produce enzymes to break lactose. Okay, but when you put this same E. coli and you just put into a media that's full of lactose, this bacteria is going to have a shift, and instead of because it doesn't have glucose, it's going to use lactose as a source of energy, of carbohydrate, and because of this, it's going to have what we call an enzyme adaptation, where instead of keep producing that same group of enzymes that would digest glucose, now it shifts to produce a lot of enzymes that are going to digest lactose to use as a form of sugar. Okay? And fun thing is that if you get the same bacteria that's in lactose media growing over there and digesting lactose, and you put that into a regular media, guess what happens? Yes, it shifts to go back to not producing enzymes that breaks lactose and start consuming regularly glucose. So also can do enzyme adaptation back to where it was before. And that's very important for bacteria, especially E. coli, because it gives to them some plasticity um, to grow in areas where it would be harder for them to grow. So it can grow, for example, in the mammary gland and can grow in the intestine as well, just because have two different source of sugars, and it can by adapting their enzymes, they can live in these two very different environments. And to understand why this happens, we have to understand that bacteria have what's called operons. So operons is a, a group of genes that are all packed together into a package, and they use these operons to produce a bunch of proteins that are going to be important to a specific thing. So, for example, we have one operon called lac operon. That's the one that we're going to be studying here. The lac operon produces a lot of different proteins that are going to be used to digest lactose. So it's going to be producing beta-galactosides, lactose permease, galactoside transactylase. So it's going to produce a lot of different proteins that are going to be important for the bacteria to use lactose as its source of sugar. Okay, and in this operon, also going to have structures to regulate and promote and regulate directly the operon, and it's also going to be regulated by regulatory genes that are outside the operon. Okay, and then we have to remember about our last class, last week actually, where bacteria, they are notoriously known to have genes that are polycistronic, means that I'm going to have an ATG, then here I'm going to have another ATG, and then here another ATG, and just at the end of it, in the terminator, is where we're going to have the TAA as termination codon. Okay. These genes, these operons, are highly regulated and they respond to the environment. So they can respond to environment, external environment, where it lacks certain nutrients or have a lot of that in nutrients, or they can respond to an internal environment. Well, usually when they're running out of a molecule, but can be because they have too much of that molecule as well. So they're going to be responding to the external and internal environment usually. In the case of the lac operon, is usually responding to the external that influence the internal environment. So let's explore further this structure of the lac operon. So as you can see in this beginning, where we're going to have a bunch of regulatory properties in here, we're going to have one site called capsid, that's from catabolite activator protein. In this site, we're going to have a DNA sequence that's going to be recognized by an activator protein. So here's where the start of the transcription is going to start. It's going to bind over there and start the transcription. Okay, this region is called the UTR region, untranslated region, even though it's transcribed in the mRNA, it's not going to be translated into protein. We're also going to have what's called an operator site in here, almost at the beginning, right before the production of the gene LEC-Z. And in the operator site, I'm going to have a DNA sequence that provides a binding site for a repressor protein that's called LEC repressor. that is going to bind over here and is not going to let any of these proteins be produced. Okay. We also have a lac promoter, where it's going to sign the start of the transcription, and lac terminator, where it's going to sign the end of the transcription. And we also have at the end of it the lac I gene that's going to produce the lac repressor that's going to bind into the operator site in case it requires. It's one way of controlling or regulating the lac operon into producing proteins to digest lactose. So in general, the lac operon is mostly regulated by a negative control. That means that if there is no lactose environment, this gene, this operon, is going to be repressed under the activation of the lac repressor that's going to be always connecting to the lac operator, resulting in a lack of all the proteins that should be produced. So that's how it is most of the time during the bacteria's life. If there's no lactose, there's no need for it, so it's always repressing this gene. Okay? If there is lactose in this environment, the lactose is going to bind into the lac repressor. By binding to the lac repressor, it no longer is going to be connecting to the lac operator. And by no longer being connected or bonding with the lac operator, all these enzymes are going to be transcripted and translated into enzymes to use lactose. Okay? Easy, right? So in the lac operon, if there's no lactose, it is repressed. If there is lactose, it's no longer repressed. Another way of also regulating the metabolism of lactose in bacteria is by glucose. So glucose is going to be connected to the AMP cyclic. That's going to be helping the CAP or CAP recruit RNA polymerase. And in general, what you guys have to remember is that if there is glucose in abundance, that bacteria is not going to produce any enzyme to digest lactose. Okay? Because of the cap interaction. So for example, here I have glucose and I don't have lactose, so the cap is not going to be connecting anywhere, but it's going to have that process that we already explained. So the gene is going to be repressed. And if there is lactose and glucose, the cap also is not going to be connecting here because still have glucose. So that bacteria is still not going to produce anything to digest lactose because it doesn't need it. Okay. But if there is lactose and no glucose, the cap is going to be connecting here. So if there's no glucose and there is lactose, or even if there's no lactose, there's just glucose, the cap is going to be connecting to the cap site, and that's going to lead to a series of events that's going to allow the production of enzymes from the lac operon to digest lactose. So in other words, if there's glucose, regardless, the lac operon is not going to be is still going to be in their repressive operation. It's going to be inhibited. It's not going to produce anything to digest lactose. If there is no glucose, there's just lactose or nothing, then the operon is going to be in activity because It's going to be trying to get whatever it can as a source of sugar to have energy. Okay? So here is just an example to get more visual. So when there's lactose outside, the lactoperon is gonna uh, no longer be repressed. This is going to allow a bunch of enzymes to be produced, and it's going to allow the lactose to enter the bacteria cell. It's going to generate a bunch of energy. As soon as it finishes the lactose as the main source and start having glucose as the main source, the lactoperon is going to be inhibited and it's going to go back to digesting glucose. Okay. Second example here is the regulation of the trip operon. The trip operon is the conjunct of genes that are going to be involved in the synthesis of tryptophan. You guys might heard about this amino acid tryptophan is one that people are always looking for because it makes you calmer, it helps you go to sleep, and I even put here people they say to have like a turkey sandwich with milk before sleeping because that's rich in tryptophan, so helps you sleeping. And here just showing that there are a bunch of other foods that are way richer than turkey with tryptophan. Okay. Tryptophan is also regulated by repressor protein, similar to LAC, but also in the opposite direction. So when there is too much, it's repressed, different from the lactose. Okay. And also there is another regulation of tryptophan by attenuation that I'm going to be explaining in the next few slides. So the first way of how the trip operon works is by a repressor that's going to bind into the mRNA, no longer allowing the production of this amino acid, the tryptophan. Okay? So when there's too much tryptophan, this repressor protein is going to connect into the UTR, 5' UTR, and is no longer going to allow the rest of the mRNA to produce this, to be expressed. So all these other genes are no longer going to be expressed. Another way of how this operon works is through attenuation. So there's the same thing here. So the trip operon is always working, always produces tryptophan. When there's too much, there is an attenuator sequence here in the 5' UTR, and this attenuator sequence is going to be it's going to detect the amounts of tryptophan, and if there's too much, this beginning of the mRNA is going to be folded in a way that's not going to allow further production downstream. If there is not too much, it's going to fold in another way that's going to allow the gene expression of all the other genes downstream. So let's understand how this works. So in attenuation, the transcription occurs, but it's terminated at the attenuator sequence. That's in the 5' UTR region. As I said, if there's high tryptophan, describe only this region, not going to be transcribing the actual enzymes that are going to allow the production of tryptophan. but there is low tryptophan, it's going to transcribe all downstream genes. So let's see this video that helps elucidate. So, in other words, if there is no stalling, just go too fast and it's going to be connecting in this region. That way it's going to inhibit the production of tryptophan. And if there is stalling, which when it has low tryptophan, it's going to be connecting this region here, and this is going to allow the production of tryptophan. Okay, very clever enzyme. You can see that this part of the operon, this UTR, is highly complementary. So that's why it's easy to be folded into these hairpins. And lastly, there's the translational and post-translational regulation. So the transcriptional regulation is the predominant way to regulate gene expression and therefore the proteins at the end of it. But in a smaller but also important way, we also have translational regulation that's going to be regulating at the level of translation, usually by riboswitches that we're going to be talking more. And there are also post-translation regulation, that's regulation at the protein level by enzymes. Usually it's very quick and very focused. It's also important, but we're gonna be talking not that much about it because it involves a lot of biochemical part of it, and since our class is on genetics, I don't think we can take advantage of this biochemical part that much. So if a protein binds in a ribosome or mRNA, it's going to impact the translation of that mRNA, correct? So these translational regulatory proteins, they're going to be binding into the mRNA. And if they repress the translation, we're going to call these proteins as translational repressors. There are also some that are going to be binding into the Shine-Dalgarno sequence or blocking the Shine-Dalgarno sequence in a way to destabilize the mRNA. That way cannot fit very well in the ribosome and it's not going to be translated. And in sometimes the bacteria genome can also produce what's called an antisense RNA that's going to be connecting to the mRNA, and that way is going to be inhibiting the production of that mRNA as well by producing a structure that's going to easily conjugate with the mRNA and is going to be for having a bond with these two molecules that's not going to allow a bond with the ribosome in order to have translation. And riboswitches are something very interesting that was covered in the last 20 years. So it's an element within the mRNA in the usually in the 5' UTR. They're very common bacteria, and what happens is that it's going to regulate that expression of translation by folding, and when it folds, it allows the connection with ribosomes to start the translation. And this folding happens by binding with metabolites or metal ions, and that's how it is regulated. It's called riboswitch just because it's like a switch, can be turned on or turned off. Okay, as I said, it's a very common way in bacteria to use riboswitch as a mechanism to regulate translation.